Welcome again. My name is Evans Matuga, your usual biology teacher, a practitioner of biology paper 1 with the normal experience on the same paper. Now, before we start our lesson, I want to believe that where you are continuing to obey the government rules and regulations of keeping safe, staying at home, and sanitizing yourselves. Well, we hope that soonest we will overcome. God is with us, and continue praying for Kenya. Now today I want us to dissect a very important aspect in Form 1, which is known as cell physiology. We are going to tackle an aspect known as diffusion. Diffusion is a very important aspect that has always featured in examinations, but students confuse it with osmosis. So I want us to concentrate first on diffusion, and then our next episode will be on osmosis. Before we understand what diffusion means, it's important that we know that this diffusion you are being taught in class all this time happens in real life situations. Where you are by now, for example, at your own homes, you are cooking ugalai, you are cooking mboga, and the smell that comes out of that ugalai or mboga is through this process called diffusion. Some of you get out a lot of gases known as fat air, and these gases can be smelling so badly. How did they reach the other person? It is through the process of diffusion. Some of you use perfumes where you're at home, and those perfumes, for you to smell a perfume from another person, it is through a process known as diffusion. So, this diffusion has been happening at and around your homes, where you are. If you are able to smell that a particular room is so stuffy, the process by which you did that is diffusion. So that is why we say that biology is a living subject. What we are seeing around, what you are smelling around, is about diffusion process. So now, what do you think this diffusion could be? It should be a process that is for a fact. And then this process, how does it happen? I want to ask you a question. For example, tomorrow, you can spray a perfume at the corner and then go to the next corner. After a few minutes, you will smell the perfume. But where do we have a higher concentration? It's where you had sprayed it. So where you had sprayed that perfume is higher concentration, and where you went to stand and try to get the smell coming is a lower concentration. So therefore, the movement must have happened from higher concentration to low concentration. And therefore, we have to define diffusion as a process by which molecules, oblique particles, move from a high concentration to a low concentration area, oblique region. So that is how simple we can understand these concepts and define them. And that becomes the definition of diffusion. Once we've understood that is how diffusion can be explained, this diffusion, we can carry out experiments both in the lab and at your homes. I will start with the laboratory because that is where I am based most of the time. Then I'll take you to your homes and see what experiments can you carry out in relation to diffusion. In our laboratory, one of the experiments that we have always performed on diffusion is an experiment that appears like this one. If you check on this diagram, this is a whisking tubing. A whisking tubing is, of course, an artificial form of cell membrane. I told you that in Form 1, there is a cell, which is the basic unit of life, and that cell has got a cell membrane. So this is a Fisking tubing, and of course you know its properties to be semi-permeable, so it can allow selective movement of substances in and out. So a Fisking tubing is of course an artificial form of a cell membrane. So therefore, if I want to carry out diffusion using a Fisking tubing, this is what I will do. This is a beaker, and this beaker contains iodine solution. So I'll need some materials which include the Fisking tubing. I will need a beaker. I will need iodine solution. I will need starch solution. And that is enough, of course, and threads so that I can close this Fisking tubing after putting starch inside. And then now wait after some time to see what happens to that solution. Now listen very carefully. When I tell you that a starch solution is what you've always known. If you look at the unga you are using at home to cook, that is starch. If you look around the unga you are buying from the supermarket, if you're buying, that is starch. So maize contains starch. Even the raboni, the sweet potatoes and the Irish potatoes, all those potatoes, whether sweet or not sweet, all of them contains starch. And now, how as a biologist can I prove that that unga you brought me from the shop is the real unga? It's containing starch. As a biologist, we have a chemical known as iodine solution, which has a color and it's brown in color. And this, if I take a reagent of iodine solution and of course take a solution of that unga you brought me from the shop and drop some iodine solution inside, the brown color turns to blue black, blue black, blue knife black. So two colors. They are not blue, they are not black. So they are in between, intermediates. So I shall have proved that this is indeed starch. So basing that statement on our experiment on diffusion, this is our Fisking tubing that we have here. And this Fisking tubing contains starch inside. And outside here, we have iodine solution. Remember, the Fisking tubing has got small pores, so that it can allow some substances, selective substances, to move in and out. In this case, between iodine and the starch, look at your unga at home and look at this iodine here. Which one is able to move in or which one is able to move out? Is it starch to move out or it's iodine to move inside? You look at which one is smaller. So the one which is smaller between iodine is a solution, just a liquid. And starch, it's a component which contains unga that you know. So you will find that unga is larger than the iodine. So therefore the iodine will move into the starch from outside to the inside. Did you get that? Once that has happened, there is an observation you are supposed to make in this experiment. And the observation you will now be able to make, because inside here we have starch, is that the inside of the Fisking tubing, the color turns blue-black. That is to show that diffusion has taken place. And the factor we are testing here is, of course, the size of the molecules. Which one is larger than the other? That is why iodine is able to move from outside to the inside. And that marks the end of the first experiment of diffusion in our laboratories. I'm coming to your homes. You wake up every day and of course you use tea and milk, and you have to make your own milk, and you are using mint tea leaves. Before you can start that experiment, cooking your tea using milk, while you are pouring the milk, go and try that experiment. The water was, of course, colorless. And while you now start pouring that milk, it starts to turn white. That is diffusion. I want you to now make strong tea. Take 2 glasses, that both of them and those 2 glasses to be containing water. And the first glass should have hot water. Another one should have, of course, cold water. Then use tea bags. Drop 1 tea bag to the cold water and drop another tea bag to the hot water. Tell me tomorrow which one turns brown faster. Which one makes a strong tea? In other words, faster is the one which has hot water. And that is to explain the temperature affects the rate of diffusion in that manner. And that is an experiment of diffusion you've been doing at home. The next one I'm going to take you is at home. Some of you are farmers and of course are bee farming farmers. And for bee farmers, it's not necessary that you must have become a bee farmer. You've been stung once, not twice, by a bee maybe. And you'll wonder, how do bees know that you've been stung and the rest now follow you? When one bee stings somebody, they of course leave you with a chemical known as pheromone. The bee is able to detect a pheromone through the process of diffusion. In fact, after a very long distance of kilometers, it can be able to sense that there is a smell of pheromone. There's a lot of story about bees and the queens, how they know and how they send the workers on how to get to their place, and of course they follow them after knowing that they have got a place to stay. That is about bees, and of course is the use of pheromones. So that is how diffusion can be very interesting, and therefore there are factors which affect the rate of this diffusion. And one of them we have already mentioned is temperature. Number 2 is the size of molecules. Number 3 is thinness of the membrane. Membrane is that Fisking tubing we are talking about. How thin is it? The next one is the type of medium that we are going to use. Surface area. Of course, surface area to volume ratio and many more. You will understand those aspects. But for temperature, I need to explain that. If you are going to perform an experiment using 2 glasses, one containing cold water and another one containing hot water, the one containing hot water, and then you placed a tea bag there and it turns brown faster, you have to tell us why. It's because high temperatures increase kinetic energy. The term kinetic energy is what we mark in your cases here. So you have to, of course, be specific to these points. And those are the factors that are affecting the rate of diffusion. Diffusion has a role in real life situation, including gases exchange of carbon dioxide and oxygen. So you must, of course, master these things and know where questions can be set from. And it is important to know those areas. See you in the next episode on osmosis.